Somewhere on a reef right now, an animal the size of your hand is loading a weapon. Its arm will accelerate faster than a bullet leaving a gun barrel. Underwater, where water should make that impossible. High-speed cameras show a crab shell splitting open. Then the water around it flashes white. That is the part people miss. This is not a punch. No muscle on Earth contracts fast enough to do this. And the strike is only half of what hits. The water gets struck twice, which raises an uglier question: why doesn't the weapon destroy the animal holding it? Start with the muscle, because the muscle is where the whole explanation falls apart. Muscle is a chemical machine. It works by sliding protein filaments past each other, and that sliding has a speed limit built into the chemistry itself. Push a muscle to shorten faster, and the force it can produce collapses. Push it further, and it produces almost nothing at all. Every animal that has ever lived is stuck inside that trade. You can be strong, or you can be quick. And the harder you lean toward one, the more you give up the other. A muscle that has to move a limb through water, dense, heavy, resistant water, 800 times the density of air, is fighting that trade with the odds already stacked against it. So when researchers first pointed high-speed cameras at a smashing mantis shrimp and started counting frames, the footage did something inconvenient. It refused to agree with the muscle. At normal video speed, there is no strike. There is an animal sitting still in a hole, and then there is a broken snail, and nothing in between. Your eye receives no event. The camera at standard frame rates receives no event either. To get anything at all, you have to run the recording at thousands of frames per second, and even then the entire motion occupies a handful of frames. In smashing species, the strike is over in under 0.003 of a second. In some measurements, the limb reaches peak speeds, somewhere in the range of 14 to 23 meters per second. And those are lab numbers from particular species, under particular conditions, not a universal figure stamped on every mantis shrimp in the ocean. Reported peak accelerations run above 10,000 times the acceleration of gravity. Sit with that for a second, because the number is easy to skim past. 10,000 G. Underwater. A fighter pilot blacks out around 9 G. A car crash peaks in the tens of G. This animal routinely pushes a piece of its own body 3 orders of magnitude beyond that, in a medium that fights every millimeter of the motion, and then does it again the next time it gets hungry. Now do the physics the animal has to do. Drag in water does not rise gently with speed. It rises with the square of it. Double the speed, and you roughly quadruple the resistance. So the faster that limb tries to move, the harder the ocean pushes back, and the pushback grows far faster than the effort does. Meanwhile, the muscle that is supposed to be doing the pushing gets weaker the faster it tries to contract. 2 curves running in opposite directions. One is the resistance climbing, one is the available force falling. Somewhere they cross, and the crossing point is nowhere near 20 meters per second. There is no version of this where muscle wins. Not bigger muscle, not better muscle. The limit is not about size or training or being a particularly aggressive crustacean. It is a limit on power. Energy delivered per unit of time. And the animal simply does not have a tissue capable of delivering that much power in that little time. Which means the power was never produced during the strike at all. It was produced earlier, slowly, and stored. That is the trick, and once you see it, the whole animal reorganizes in front of you. The mantis shrimp does not have a fast muscle. It has a slow muscle attached to a spring. The muscle takes its time, a leisurely fraction of a second, geological by comparison, and pours energy into a deformable structure in the arm. The energy sits there, compressed, waiting. Then it is released all at once. The total energy has not changed. What changed is how long the release takes. Squeeze the same energy into 1/10 of the time, and you have 10 times the power. Squeeze it into 1,000th, and you have something that no biological tissue could ever generate directly. That is why the footage looks fake. You are not watching a muscle contract, you are watching a latch let go. And that raises the question that actually matters, the one that turns this from a fun fact into an engineering problem. How do you hold a loaded spring inside a living body without it going off? Because a spring is easy. Springs are everywhere in biology. Tendons are springs. Insect wings run on springs. The arch of your foot stores and returns energy every time you take a step. Storing elastic energy is one of the oldest tricks in animal design. What is not easy, what is genuinely hard, is holding a large amount of stored energy motionless, indefinitely, in warm, wet tissue, and then releasing all of it in microseconds, on command, aimed, without the whole assembly tearing itself apart. That problem has a solution, and the solution is buried in the second segment of the arm. The structure at the center of it is called the saddle, and the name is literal. Look at the meris, the thick upper segment of the raptorial limb, and there is a small curved shell of exoskeleton on its upper surface that is shaped exactly like a horse saddle, curving down along one axis and up along the other. Engineers have a name for that shape. It is a hyperbolic paraboloid. It is the same geometry used in certain concrete roofs, and it is used there for a specific reason. That double curvature lets a thin shell take enormous compressive load without buckling sideways. A flat plate under compression folds. A saddle-shaped shell under compression stays put and stores the strain. The material choice is stranger than the shape. The saddle is not one substance. It is layered, and the layers are doing opposite jobs. The outer layer is heavily mineralized, a stiff, brittle, ceramic-like material, closer to bone or tooth in character than to ordinary shell. The inner layer underneath it is fibrous and organic, closer to a rope or a bundle of cable. Now think about what happens when the saddle bends. The outside gets squeezed. The inside gets stretched. Ceramics are excellent under compression and terrible under tension. They crack. Fibers are excellent under tension and useless under compression. They buckle. The saddle puts the ceramic exactly where the compression is and the fibers exactly where the stretching is. So each material only ever experiences the load it happens to be good at. That is not a general-purpose piece of shell. That is a component. It reads like something drawn on purpose. And the saddle is not working alone. It sits inside a linkage, a set of rigid exoskeletal elements connected at pivots, including a wedge-shaped structure sometimes called the Merrill V, and supporting bars running along the underside of the segment. The whole assembly behaves like a mechanical linkage of the kind you would find in a machine. Input at one end, motion transformed and amplified at the other. Muscles contract, the linkage compresses the spring, and the geometry converts a slow, powerful, short pull into a fast, wide rotation of the club. But none of that fires until something lets go. The release depends on small, hardened plates, latch structures, that physically hold the folded limb in the cocked position while the muscle loads it. This is the part that gets skipped in every short version of the story, and it is the most interesting part. The animal is not just building force, it is managing a countdown. Load, hold, aim, release. The energy goes in over a comparatively long stretch of time and comes out in a burst measured in microseconds. And the only thing standing between those two states is a latch a fraction of a millimeter across. Watch a smasher hunt, and the sequence becomes visible in its behavior. It does not charge. It positions. It walks a snail into range with its front limbs, turns it, adjusts, and holds. The eyes move independently, tracking, measuring. Only then does the arm cock. And if you are watching at normal speed, the cocking is the only part you actually see. There is a small tightening, a shift of posture, an animal that suddenly looks compressed, then nothing. Then damage on the shell that was not there a moment ago, and that tells you something about the cost structure of the whole system. A misfire is not free. Every strike burns a full loading cycle, and a loading cycle takes time and energy. An animal with a limitless weapon would swing constantly. This one aims first. That is not restraint out of temperament. That is an animal that cannot afford to waste a shot, behaving exactly the way you would expect a creature with a limited magazine to behave. So the speed is accounted for. Slow muscle, stiff spring, small latch, big release. The mechanics close, except the mechanics do not close because there is still a problem sitting in the water and nobody has dealt with it. The problem is the water itself. That limb is moving through a fluid that cannot get out of its own way fast enough. Water is not compressible in any useful sense. You cannot squash it. When something moves through it slowly, the water simply flows around the obstacle and closes back up behind. When something moves through it very fast, the closing up part starts to fail. The fluid behind the moving object cannot rush in quickly enough to fill the space being vacated. The pressure in that gap drops. And when pressure in a liquid drops far enough, the liquid does not simply thin out. It changes phase. It boils. Not from heat, but from the absence of pressure. And a pocket of vapor tears open inside the water. That is cavitation. And it is not a curiosity. It is the reason ships lose propellers. Spin a steel propeller fast enough, and the low-pressure side of each blade starts generating these vapor pockets. They form, they travel, and then they meet water at normal pressure again, and they collapse. The collapse is not gentle. The surrounding water rushes inward to fill the void, accelerating as it goes, and it slams shut with a violence completely out of proportion to the size of the bubble. Do that a few million times, and the steel starts to look like it has been eaten, pitted, cratered. Metal removed from a solid blade by nothing but water changing its mind about what phase it wants to be in. Naval engineers have been fighting cavitation for over a century. It destroys pumps. It erodes turbines. It limits how fast a submarine can turn its screws before it starts announcing itself to every hydrophone in the ocean. And a hand-sized crustacean on a coral reef generates it on purpose. When the club swings, the trailing face of that limb outruns the water. A vapor cavity opens right at the strike zone, between the club and the shell it is about to hit. Microseconds later, that cavity collapses. The measurements are what turn this from an interesting side effect into the actual weapon. In work on the peacock mantis shrimp, each strike produced not one force peak, but two, separated by roughly 400 microseconds. The first peak is the club hitting the target, with impact forces in that study running from around 400 newtons up to about 1,500. The second peak is the cavitation bubble collapsing, and it reached values around 500 newtons. So the second hit is not a rounding error. In that work, the cavitation force typically ran at about half the limb's impact force, and under some conditions the ratio ran far higher, with cavitation forces reported to exceed the limb impact substantially. The acoustic signature of the collapse is brutally short, on the order of tens of microseconds. Read that again in terms of what it means for the prey. The snail is not struck once. The snail is struck, and then, before the shell has finished responding to the first blow, it is struck again by the water. Two loading events, back to back, faster than any material can relax between them. Shell is not bad armor. Shell is very good armor against slow, sustained crushing, which is what most predators deliver. A crab squeezing, a fish grinding. Shell is much worse at handling repeated high-rate impacts because brittle materials fail through cracks, and cracks take time to be resisted but almost no time to travel. Hit it once, and you start a crack. Hit it again 400 microseconds later, while the crack is still open and the structure is still ringing, and you drive that crack the rest of the way through. There is a detail buried in this that changes the animal from a hammer into something much harder to defend against. The near miss still works. If the club swings and does not quite make contact, the cavitation still forms because cavitation is generated by the speed of the limb through water, not by the collision. The bubble still collapses. The shock wave still arrives. A prey animal that dodges a mantis shrimp strike has not necessarily escaped the strike. It has only escaped the club. A snail can be cracked, stunned, or killed by a blow that technically missed it, which means the effective kill radius of this animal is larger than its reach. There is a zone around the strike where the water itself becomes the weapon, and nothing in the visual scene tells a prey animal where that zone ends. That alone would justify the reputation. But there is something else happening inside those collapsing cavities, and it sits right on the boundary between confirmed and inferred, so it needs handling with care. When a cavitation bubble collapses, the inward rushing water compresses whatever gas and vapor is trapped inside. Compression heats gas. Fast compression heats it fast, because there is no time for the heat to leak out into the surrounding liquid. That part of the physics is not controversial. What is genuinely startling is how far it goes. In snapping shrimp, a different animal entirely, a small crustacean that snaps a specialized claw shut to produce a cavitation bubble as its hunting weapon, researchers found that the collapse emits a brief flash of light. The phenomenon picked up a name of its own, borrowed from the study of sound-driven bubbles in the lab. A short, extremely brief pulse, on the order of nanoseconds, coming out of a collapsing bubble in seawater. From the light and from the models, estimates of the temperature inside those collapsing cavities run into the thousands of Kelvin and beyond, into figures that get compared to the surface of a star. That comparison sounds like hype until you remember what it applies to: a volume smaller than a grain of sand for a duration shorter than a camera flash. That temperature is not measured with a thermometer. Nothing could survive being a thermometer in that position. It is inferred from spectra and from bubble collapse models, and researchers have argued about the exact numbers and the exact mechanism for decades. Treat those figures as estimates, not readings. For mantis shrimp specifically, the mechanical side is solid. The bubbles are there. The collapse is measured. The second force peak is real. The light is the part where the hand should stay hovering over the caution button. Faint emission associated with these collapses has been reported under laboratory conditions, and the underlying physics belongs to the same family of phenomena. But nobody is filming this casually on a reef, and the mechanism remains an argument in progress rather than a closed question. So here is the careful version, and the careful version is still worse than the myth. The animal is not making a tiny sun. The animal is making a region of water in which pressure, temperature, and energy density briefly go somewhere they are not supposed to go, and it is doing that as a routine part of eating lunch. Whether or not a photon escapes is almost beside the point. The point is that the energy density is high enough for the question to be reasonable, and that is exactly where the problem turns around and points back at the animal. A collapsing bubble does not care which side it is on. The cavity forms between the club and the target. When it implodes against both of them. The same process that pits ship propellers is happening a fraction of a millimeter from the striking face of a living limb. Not once, but every single time this animal feeds for its entire life. That limb should be destroyed. Not slowly, not eventually, immediately. Take any engineered material you like and put it on the striking end of this. Steel erodes under cavitation. Ceramic is hard but brittle, so it survives the compression and shatters on the crack. Metal is tough but soft, so it survives the crack and deforms into uselessness. The classic engineering trade sits exactly here. Hardness against toughness. Hard materials resist being dented and fail catastrophically once a crack starts. Tough materials absorb cracks and fail by going out of shape. You normally cannot have both. The Mantis Shrimp Club has to have both. Plus, it has to be light. Plus, it has to be grown rather than manufactured. Plus, it has to keep working while soaked in seawater and attached to living tissue full of nerves. So look at what it actually is. The striking face is not shell in the ordinary crustacean sense. The outermost layer is a thin coating, roughly 70 micrometers thick, thinner than a sheet of paper, made of a mineral in the calcium phosphate family, the same broad chemistry that builds bone and tooth enamel. Measured hardness for that coating comes out extraordinarily high for a biological material, in the range of tens of gigapascals. That is the part that meets the snail. Its job is to not dent, but a hard, brittle skin on its own is a liability. Hit it hard enough, and it cracks straight through, and the crack keeps running into whatever is underneath. So the layer beneath it does the opposite job. Under the coating sits a region, a few hundred micrometers deep, built from mineralized chitin fibers arranged in a herringbone pattern, fibers laid in alternating tilted bands, the way you would lay bricks or parquet flooring if your specific goal was to make it hard for a crack to find a straight path. That is exactly the function. When impact energy enters this region, it does not produce one clean fracture. It produces a diffuse mess of tiny cracks that start, wander, run into the next fiber orientation, and stop. Notice what that means. The club is not preventing damage. It is not too tough to break. It is built to break in a specific, controlled, distributed way. To take the damage and spread it sideways, instead of letting it drive inward. That is a completely different philosophy of armor from the one most people carry around. We think of armor as a wall. This is closer to a maze, and underneath the maze is the part that makes materials scientists go quiet. Deeper into the club, the fibers stop lying flat and start twisting. Layer after layer of chitin fiber bundles are stacked with each layer rotated slightly relative to the one below it, so that following the fiber direction through the thickness of the club traces a slow spiral, like a spiral staircase running down through the material. This arrangement has a name in materials science, a Bouligand structure, and versions of it show up across the biological world, in fish scales, in beetle shells, in bone. It is one of nature's standard answers to the problem of making something crack-resistant out of fibers. But in this animal, the spiral is not uniform. The pitch changes through the depth. The rotation angle between neighboring layers is graded rather than constant. And that grading turns out to matter for a reason that has almost nothing to do with cracks. It has to do with sound. When something strikes a solid, the impact does not just push. It sends waves through the material. Vibrational energy racing through the structure at the speed of sound in that medium, bouncing, reflecting, interfering. Two broad kinds matter here. Pressure waves, which travel by squeezing and stretching the material along the direction of travel, and shear waves, which travel by sliding layers of material sideways past each other. Biological tissue does not mind pressure waves much. Tissue is mostly water, and water handles compression well. Shear is the problem. Soft, wet, living tissue tears under shear, nerves, muscle, the delicate machinery inside the limb. All of it is far more vulnerable to being slid apart than to being squeezed. Work on the club structure has argued that the graded spiral region behaves like a filter, not a wall, not a cushion, a filter, in the same sense that certain engineered structures block particular frequencies of sound while letting others pass. The repeating, progressively rotated layers create a periodic architecture, and periodic architectures interact with waves in ways that depend on wavelength. Waves whose wavelength matches the periodicity of the structure get scattered, reflected, and canceled. Waves that do not match pass through. The finding was that this region appears to selectively reject high-frequency shear waves, exactly the component most likely to shred the soft tissue behind the armor. So the club is not one device. It is at least three stacked. A hard skin that refuses to dent, a herringbone layer that captures and scatters cracks, and a graded spiral region that filters the vibrational energy of the animal's own strike before it reaches anything alive. The caution here has to be stated plainly, because this is recent work and it comes with limits its own authors were direct about. The wave analysis came from simplified two-dimensional models. The real club is three-dimensional and considerably messier, and the researchers were explicit that fuller modeling and experiments in actual water are needed before anyone claims the picture is complete. What is not in doubt is the architecture. The layers are there. The grading is there. The question still being worked out is precisely how much of the protection comes from filtering versus from the other mechanisms operating at the same time. That uncertainty does not weaken the story. It sharpens it. We are looking at a structure whose function we have not finished decoding, on an animal that has been using it, without needing to understand it for a very long time. This is also the part where the mantis shrimp stopped being purely a biology subject and became an engineering one. Design cues taken from this club have been fed into prototype composite materials, layered fiber composites built with deliberately graded, twisted stacking sequences, tested for impact resistance with applications aimed at aerospace structures and protective equipment. The logic is straightforward. If you want a panel that takes a hit without shattering and without letting the shock through to what is behind it, you could keep making it thicker and heavier. Or you could copy the arrangement that a crustacean arrived at through a few 100 million years of not having any other option. There is something slightly humbling about the direction of that transfer. This is not people making better armor and then noticing an animal that resembles it. This is people looking at an animal, failing to explain how the material survives, taking it apart layer by layer, and finding a design they had not thought of. And the reason that design has to be quite so good only becomes clear once somebody counts the strikes, because the single-punch kill is mostly fiction. Work that tracked smashers handling snails found something the highlight reels never show. The animals did not deliver one decisive blow. They delivered dozens. In some trials, hundreds. Across that study, the average ran to something like 80 odd strikes on a single snail, with individual trials ranging from under 10 to several hundred, which changes the scene completely. A mantis shrimp working on a snail is not executing. It is demolishing, returning again and again to a shell that resists, hitting, checking, repositioning, hitting again. And it does not hit randomly. The same work found the strikes were aimed. On round, globular shells, the animals concentrated their blows on the aperture, the opening, the one place where the structure is already interrupted and where a crack has somewhere to go. On tall, high-spired shells, they shifted their targeting toward the apex instead. The middle of the shell, the broad, reinforced whorls, was almost never chosen. That is not swinging. That is targeting. The animal is not applying force to a shell. It is applying force to the weakest available feature of one particular shell shape. And it changes that choice depending on what is in front of it. And now the fracture logic from earlier turns around and bites. Cracks are easiest to start at a discontinuity and hardest to drive through solid, curved, reinforced material. An edge is a discontinuity. So is a spire tip. By going after those, the animal is not being stubborn. It is picking the point where its two-part strike will do the most damage per loading cycle. Even so, the snail usually holds. Repeatedly. That is what armor is for, and it is a reminder that this arms race is not lopsided. The shell is winning most of the individual exchanges and losing only the war of attrition. So the wear problem sits in an entirely different bracket than it looked, because the club is not absorbing one impact per meal. It is absorbing dozens, sometimes hundreds, every single one of them carrying an impact peak and a cavitation collapse straight back into the same few hundred micrometers of mineral and fiber. And for all of that, the club still loses. Chips accumulate on the striking face. The mineral coating erodes. Microcracks fill the herringbone layer, which is exactly what they are supposed to do. But a crack-arresting structure only has so much room to keep arresting cracks before the region is saturated with them. The club is not eternal. It has a service life. It wears out. And a crustacean cannot repair its exoskeleton. There is no healing, no remodeling, no bone-like turnover. The shell is dead material laid down outside the body. Once it is damaged, it stays damaged. So the only way to get a new club is to grow a new one. And the only way to grow a new one is to leave the old one behind entirely. This is where the story turns, because the animal we have been building, the ceramic-tipped, spring-loaded, cavitation-generating problem, periodically becomes something else. It molts. And when it molts, it becomes soft. Molting in a crustacean is not like a snake shedding skin. A snake under its old skin already has a new one, functional and ready. A crustacean has to first split and abandon its entire external skeleton. The shell, the leg segments, the mouth parts, the eye surfaces, the lining of parts of the gut, all of it in one piece, stepped out of like a body-shaped suit of armor. What emerges is the same animal at a slightly larger size, wrapped in a new cuticle that is not hardened yet. And unhardened is not a mild word here. Fresh crustacean cuticle is flexible and thin. The animal that just molted does not have a rigid skeleton at all in the ordinary sense. It is holding its shape mostly through internal fluid pressure, the way a garden hose holds its shape when the water is on. It is briefly closer to a soft-bodied animal wearing a wrapper than to the armored thing it was yesterday. Now put the weapon back in the picture. The strike works because the spring is stiff and the latch is rigid and the club is hard. Take away the stiffness and the whole mechanism stops functioning. A soft saddle cannot store the energy. A soft latch cannot hold it. And even if it somehow could, a soft club driven at those speeds would not break a snail. It would break itself. There is a documented ordering to how the hardening proceeds after a molt, and it is not random. The raptorial appendage, specifically the segments that carry the spring and the club, is among the last to become rigid again. The animal gets its body back before it gets its weapon back, which produces one of the strangest situations in the ocean. For a stretch of days, the most mechanically extreme predator on the reef is completely unable to hit anything. It is not slower. It is not weaker. It is disarmed. The crabs, the octopuses, the fish that would normally keep their distance are now looking at something they could simply eat. And the mantis shrimp knows it, or at least, it behaves exactly as if it does. Studies of newly molted stomatopods found that they do not fight. They do one of two things when threatened in their burrow. They flee, abandoning the cavity, which costs them more than it sounds like. Or they bluff. Bluffing in this animal means performing the threat display, the morale spread, the raptorial limbs swung outward and forward, opening the arm segments to present the brightly marked inner surfaces, the whole body squaring up in the burrow entrance. In a hard-shelled individual, that display is a promise. It says, "Come closer, and I will hit you, and you know what happens when I hit you." In a freshly molted individual, that display is a lie. There is nothing behind it. The limb being brandished cannot fire. If the intruder ignores the display and pushes in, the bluffer has no second option. Think about what has to be true for that behavior to exist at all. It only works if opponents usually retreat from the display without testing it. It only pays if the cost of being caught bluffing is lower than the cost of immediately surrendering the burrow. And it can only persist evolutionarily if it is not used too often, because a signal that is frequently false stops being obeyed. This is a system where honest signaling and deception are balanced against each other, in an arthropod, over a hole in the reef. And that same research pointed at something else that makes the vulnerability worse. If an intruder does strike a freshly molted animal on the tailplate, the armored shield these animals normally present to absorb blows, the soft telson may deform badly or tear rather than take the hit. The defensive gear does not just work less well when soft. It can fail outright. So the animal has a weapon with a maintenance cycle, and during maintenance, every part of its defensive system goes offline at once. Which raises the question that has been sitting underneath this whole time, waiting. Where does something like this hide? The answer is a hole. And the hole turns out to explain more about this animal's behavior than the weapon does. Look at a reef the way a stomatopod has to look at it. Most of what you see is useless. Open sand is death. No cover. Nothing to back into. Visible from every angle to every predator with eyes. Live coral is structure, but structure that is occupied and defended by other things. What a mantis shrimp needs is a specific and surprisingly rare object, a cavity in hard substrate, with an opening narrow enough that the animal can block it with its own body and armor, deep enough that nothing can reach in, and stable enough that it will not collapse. In rubble-dwelling smashers, that usually means a pre-existing hole in a chunk of dead coral or rock, a cavity bored out by something else, an old worm tube, a gap in the limestone. The animal does not manufacture it from nothing. It finds it, modifies it, and moves in. Now count those cavities across a patch of reef and compare that count to the number of animals that would like one. The math is not friendly. Suitable cavities are a limited resource, they are not evenly distributed, and demand for them does not go down. Every mantis shrimp in the area needs one. So does every animal that grows, because a cavity that fit you last year does not fit you this year. The population is permanently in a state of housing shortage. That shortage sets the terms for everything. A stomatopod with a good cavity has somewhere to retreat when a predator passes. It has somewhere to sit while its new shell hardens after a molt. It has a defensible position from which to ambush prey walking past the entrance. It has a place to hold eggs. Without a cavity, it has none of that. An evicted mantis shrimp in open water is not a fearsome predator. It is a soft-bodied crustacean with no cover, which is one of the more reliable ways to be eaten on a coral reef. So here is where the popular version of this animal gets the story backwards. The reputation says the mantis shrimp is violent because it is a killer. Watch the actual behavior and a different picture assembles. Hunting, for a smasher, is often not especially dramatic. It involves waiting, positioning, and delivering a single strike to something that cannot run very fast anyway. Snails do not fight back. Crabs put up a brief argument and lose. The hunting is efficient and it is over. The fighting is a different matter entirely, and the fighting is about real estate. A large share of the aggression a stomatopod displays in its life is directed at members of its own species over cavities. Intruders arrive and probe. Residents defend. Larger animals try to evict smaller ones. Animals that have just lost a fight go looking for a hole occupied by someone weaker. It is a continuous, low-grade property dispute running across the whole reef, and it does not stop, because the underlying scarcity does not stop. That reframes the title claim in a way that is less cinematic and considerably more unsettling. This is not an animal built for rage. It is an animal built for a market with too few units and no way to build more. And that creates a genuine problem, one that lands on the species rather than the individual. You have a dense population of animals that all want the same scarce object. You have given every one of them a weapon capable of shattering shell and generating shock waves, and you have made them fight each other over that object, repeatedly, throughout their lives. Run that forward. Every dispute becomes potentially lethal. Every contest risks killing one participant and injuring the other. A population that resolves its housing shortage by having its members destroy each other does not last long because the weapon is too good. Winners get damaged, losers get killed. The reproductive cost stacks up fast. Something has to give. And when you actually watch stomatopod contests, you find that something did. They do not fight the way they hunt. The escalation runs through stages, and the stages are consistent enough that they can be described as a sequence. It starts with distance and information. The animals track each other visually, those independently moving eyes swiveling to lock onto an approaching individual. There is chemical assessment too, antennae flicking through the water sampling whatever the other animal is releasing. Both are trying to answer the same question before anything physical happens. How big is this one? And can I beat it? If that does not settle it, the display comes out. The merrill spread, the same posture the freshly molted animal uses to bluff. With the raptorial limbs opened wide and the marked surfaces of the arm segments turned toward the opponent. This is advertisement. It makes the animal look larger, and it puts the weapon on visible display without using it. If the display does not settle it, they escalate to contact. And this is the part that does not behave the way you would expect. They do not aim for each other's soft parts. They do not go for the eyes, the gills, the abdomen, the joints, all of which are available and all of which a single strike would destroy. Instead, the animal being struck turns away, curls its abdomen forward, and presents its tail plate, the telson, the heavily armored rear shield, and then the other one hits it, deliberately, on the armor, while the recipient holds still and takes it. This behavior is called telson sparring, and once you know it exists, it changes what the weapon is for. Two animals in a burrow dispute exchange strikes to each other's tail armor, in turn, and the outcome of the dispute is decided by what happens during that exchange. One of them eventually breaks off and leaves, which means the strike is functioning as a measuring instrument. The telson is where this gets architecturally interesting. It is not simply a thick plate. Stomatopod tail plates carry raised ridges and keels, and beneath the surface, the structure is built to deform and absorb, rather than to be rigid. Work on the impact mechanics found that the telson behaves like a shock absorber, taking the energy of an incoming strike, dissipating a large portion of it, and springing back. Comparative work across stomatopod species has looked at how this armor evolved alongside the ritualized fighting behavior itself, which is the kind of pairing that tells you the two things shaped each other. So the animal has a weapon tuned to shatter shell, and a shield tuned specifically to survive that weapon. Not predator armor, not general purpose armor. Armor calibrated against its own species. And that is what makes the sparring work, as a signal that cannot easily be faked. You can bluff a display, posture is cheap. What you cannot bluff is the actual force delivered by your own strike, because the force comes from the size and condition of a spring that either is or is not loaded, in a limb that either has or has not hardened. When a stomatopod takes a hit on the telson, It is receiving direct physical information about the other animal's capability, delivered through its own armor. Analysis of these contests supports a mutual assessment model, both animals gathering information about each other and about the difference between them, rather than each simply fighting until its own energy runs out. The decision to retreat comes from comparison, not exhaustion. So the violence is real, it is frequent, and it is almost entirely regulated. Two animals capable of killing each other in a single motion instead run a structured exchange that ends with one walking away intact. This is the point where the title's second word has to be re-examined: most violent predator in the ocean. The strikes are real. The forces are real. But the function is closer to arbitration than slaughter. It is violence used as a currency, spent in measured amounts to settle disputes that would otherwise have to be settled by killing. And there is one more consequence of this system, easy to miss and genuinely strange. It means a mantis shrimp needs to be able to judge another mantis shrimp: size, condition, whether it has recently molted, whether it is bluffing, whether it is worth fighting. That judgment has to happen fast, often in murky water, often in low light inside a rubble field, against an opponent that is actively trying to appear more dangerous than it is. You cannot run a bluffing economy without good sensors. If displays could not be read accurately, displays would be worthless, and everything would collapse back into lethal fighting. So the eyes are not decoration. The eyes are load-bearing, and the eyes of this animal are where the story stops being about mechanics and starts being about something considerably harder to get your head around. Start with what you can see from outside, because even the outside is wrong. The eyes sit on stalks, and they move. Not the way a crab's eyes move, which is mostly a matter of raising and lowering. These rotate. Each eye can swivel independently of the other, and independently in all three axes of rotation: side to side, up and down, and around its own optical axis. That last one is the strange one. The eye can roll, twisting the whole visual field, and rotations of up to about a quarter turn have been recorded. Watch a mantis shrimp in a burrow entrance, and the movement is genuinely unsettling to a human observer, because it violates something we take for granted. Our eyes are yoked. They move together, always, and the yoking is deep enough that you cannot easily override it by trying. A mantis shrimp has no such constraint. One eye can be tracking a passing fish while the other scans the seafloor. They can also lock together and work in concert when the task requires it. The independence is not a permanent state, but a mode the animal switches into and out of depending on what it is doing. Then look at the eye itself, and it splits into three. Each eye is divided by a horizontal band running across its middle. Above the band, a hemisphere of ommatidia, the individual optical units of a compound eye. Below the band, another hemisphere, and between them, the midband, a narrow strip of specialized rows. Up to 6 of them in the most elaborate species, each row built differently from the others and from the hemispheres above and below. Here is what that division does, and it is the part most people have never heard. The upper hemisphere, the midband, and the lower hemisphere are aimed such that a single point in space can be viewed by all three at once. 3 separate optical samples of the same location, in one eye. Which means each eye on its own has depth perception. Stop and consider how unusual that is. Your depth perception comes from having 2 eyes in different positions and a brain that compares the 2 images. Take one eye away and you lose most of it. You are left with weaker cues like motion parallax and known object size. Almost every animal with binocular depth perception works this way. 2 eyes, 1 comparison. A mantis shrimp gets range from 1 eye, which means it can range find on 2 different targets simultaneously in 2 different directions, with 2 eyes operating on independent business, Now think about why an animal would need that specific capability. Go back to the strike. The whole system is built around a single release of stored energy that cannot be recalled, cannot be corrected mid-flight, and costs a full loading cycle, whether it hits or not. There is no tracking, no adjustment, no second chance within the same motion. Once the latch lets go, the outcome is already determined by the geometry set before firing. A weapon like that is worthless without precise range finding. Fire too early, and the club sweeps through water. Fire too late, and it collides at the wrong point in its arc. And the whole event unfolds faster than any nervous system could possibly correct it. So the aiming has to be complete before the release, which means everything the animal needs to know about the target's distance has to be gathered in advance. The eyes are not a strange accessory bolted onto a strange weapon. They are the targeting system that makes the weapon usable at all. But the depth perception is not what made these eyes famous. What made them famous is the midband, and specifically what is packed into it. In the upper 4 rows of a 6-row midband, the individual optical units are divided vertically into stacked tiers, and the tiers sit at different depths under different filtering material. Each tier absorbs a different portion of the spectrum. Counting the tiers across those rows gives 12 distinct spectral channels, 12 different answers to the question, what wavelength is arriving here? Counting all the specialized receptor types in the midband, including the ones dedicated to other tasks, the total runs up to around 16. For comparison, you have 3. 3 cone types, red, green, blue, and every color you have ever perceived. Every sunset, every painting, every shade you can name, and every shade you cannot, is assembled by your brain from the relative activity of 3 channels. Some of the Mantis shrimp channels extend into the ultraviolet, where you are simply blind. And the ultraviolet part carries an extra layer of refinement that shows the eye is not just accumulating receptors, but tuning them. In at least one species, the optical elements above certain ultraviolet receptors contain filter pigments, a set of chemically distinct compounds, from a family of ultraviolet absorbing molecules common in marine organisms, deposited in the crystalline cones sitting over specific facets. The filters cut into the ultraviolet range and slice it, so that receptors which would otherwise respond to a broad smear of ultraviolet instead respond to narrower, separated bands. That is not accidental. That is optical engineering, executed in pigment, to sharpen the divisions between channels the animal already had. So the setup is extraordinary, and for years, the conclusion drawn from it was obvious. 12 or more spectral channels must mean color vision beyond anything we can imagine. The claim spread everywhere that this animal perceives colors that do not exist for us, that its visual world is unimaginably rich, that our 3-cone experience is a gray shadow of what it sees. The logic seemed airtight. More channels, more color. Then, someone actually tested it. The test was behavioral, which matters, because anatomy tells you what an eye can detect, and behavior tells you what an animal can actually distinguish. Those are different questions, and they can have different answers. The experiment used a standard approach in animal vision research. Train the animal to associate a specific wavelength with food, then present it with that wavelength alongside a slightly different one, and narrow the gap. The point at which the animal stops choosing correctly tells you the finest color difference it can resolve. The results did not go the way anyone expected. The animals could tell apart colors that were far apart on the spectrum. Blue from green, green from orange. No problem. But as the 2 wavelengths were brought closer together, performance fell off far sooner than it should have. The wavelength gap they needed in order to discriminate reliably was large. Compared to a human, whose discrimination at the best parts of the spectrum is extremely fine, their performance was worse. Not marginally, substantially, across the spectrum. An animal with 12 or more spectral channels was outperformed at color discrimination by an animal with 3. That result is one of the cleanest reversals in modern sensory biology, and it is worth being precise about what it does and does not say. It does not say the mantis shrimp is colorblind, or that the receptors are useless, or that the anatomy was misread. The receptors are there, and they respond as described. What the experiment showed is that fine wavelength discrimination, the ability to say these two nearly identical colors are different, is not what this system is good at, which forces a much better question. If the machinery is real, and the obvious use of it is not what it does, then what is the machinery for? The interpretation that came out of that work is a genuine departure from how vision is usually assumed to operate, and it hinges on cost. Your color vision works by comparison. Your brain takes the outputs of your three cone types and computes differences between them: red against green, blue against yellow. And from those differences, it constructs a color. It is a calculation, and it is done for every point in the visual field continuously. With 3 inputs, that means a manageable number of comparisons. Add channels, and the number of possible comparisons climbs steeply. 12 channels, compared against each other in the same way, would demand a great deal of neural machinery and a great deal of processing time. Time is the constraint that matters here. This is an animal whose entire predatory strategy is built on a single unrecallable release, in a habitat where prey walks past a burrow entrance for a brief window and then is gone. A visual system that produces exquisite color judgments half a second late is worse than useless. The proposal, based on those behavioral results, is that stomatopods are not computing color by comparison at all. Instead, the eye scans. Those constant, sweeping mid-band movements drag the narrow strip of receptor rows across an object, and as it sweeps, the 12 channels fire in a sequence, determined by what the object reflects. The output is a pattern in time, rather than a computed ratio. The comparison that gets used for this is a barcode scanner, and it is a good one. A scanner does not analyze a barcode. It does not measure the width of each stripe and reason about it. It runs a beam across the pattern and matches the resulting signal against known signals. It answers a recognition question. Which one is this? Not a measurement question. How far apart are these two? Under that model, the mantis shrimp is not seeing more colors than you. It is identifying colors faster in one pass without the computation. It trades precision for speed and for a cheap nervous system. That reframing cuts against something almost everyone assumes without noticing, that more sensory hardware must mean richer experience. Here, more hardware appears to buy a shortcut. The animal did not build a better color computer. It built a system that avoids needing one. 2 caveats, both important, both honest. First, the scanning model is the leading interpretation of those results, not a closed case. Work in this area has continued, and some of it has found behavioral evidence pointing toward opponent-style processing in stomatopods after all, which complicates the clean version of the story. The field has been openly describing this as an area with more questions than settled answers. Second, the behavioral finding is about wavelength discrimination specifically. It does not rule out that the system is doing other things with those channels that no one has designed the right experiment for yet. But the direction of the correction is not in dispute, and it is the direction that matters for anyone who has repeated the popular version. The famous claim that the mantis shrimp sees a rainbow beyond human imagination is, at minimum, not supported by how the animal actually behaves when tested, and that leaves the last 2 rows of the midband unaccounted for, because 12 channels sit in the top 4 rows. The bottom 2 rows are doing something else entirely. They are reading a property of light that has no color at all. Light travels as a wave, and a wave has an orientation. As a light wave moves forward, its electric field oscillates side to side, And the direction of that oscillation is a real physical property, as real as wavelength, and completely invisible to you. Sunlight arriving at the ocean surface has no preferred orientation, but light that scatters off water molecules, or reflects off a surface at an angle, or passes through certain structured materials, comes out with its oscillations lined up. That is linear polarization, and it is everywhere in the sea. The underwater light field is patterned with polarization in ways that shift with depth, with the angle of the sun, with the direction you are looking. Polarizing sunglasses exist because reflected glare off water and roads is polarized, and a filter oriented the right way can delete it. You have used the phenomenon. You have never seen it. Plenty of animals detect linear polarization. Many insects use the polarization pattern of the sky as a compass. Cephalopods detect it. Several crustaceans detect it. It is unusual, but not extraordinary. What stomatopods have is extraordinary. The lower rows of the midband are built for polarization, with the receptor structures oriented so that different rows respond to different orientations of the oscillation. The animal does not merely notice polarization. It can compare orientations against each other, the way your visual system compares wavelength channels. And the way the receptors are physically arranged in the eye, it means the animal can adjust its sensitivity by rotating the eye itself, which is one plausible reason for those torsional eye rolls. Turning the eye turns the analyzer. That would already be a sensory channel most animals cannot access. But there is a second, rarer form. Some light does not just oscillate in a fixed plane. It can rotate as it travels, the electric field corkscrewing around the direction of propagation, either clockwise or counterclockwise. That is circular polarization. It is common in laboratories, in optical instruments, in specialized filters. It is rare in nature, and, until relatively recently, nothing in the animal world was known to detect it. Stomatopods do. The work that established this brought together several independent lines of evidence rather than resting on one. Behavioral responses, electrical recordings from photoreceptors, the optical anatomy of the receptor structures, and the physical design of the reflective signals on the animals themselves. Certain receptor units in the eye act like a component optical engineers call a quarter-wave retarder, a structure that converts circularly polarized light into linearly polarized light, which the underlying receptors are already equipped to read. And crucially, the animals were shown to behave differently toward the two rotational directions, which is the difference between an eye that is physically capable of the trick and an animal that actually uses it. Then the reason it matters: parts of some stomatopods' own bodies reflect circularly polarized light. Specific patches, on specific body regions, on specific species. Surfaces that look ordinary in normal footage and light up when viewed through the right filter. That leaves an image that is hard to put down. There are animals on the reef right now marked with patterns that almost nothing else alive can perceive. The leading interpretation is that this functions as a signaling channel, possibly tied to mate choice and sexual display. A way of communicating that other species on the reef cannot read. A private channel in the middle of an environment saturated with eyes. And the logic behind that hypothesis is genuinely good because it solves a real problem. Signaling is dangerous. Any signal loud enough to reach the intended receiver is also available to everything else that can detect it, including predators. A bright color patch that attracts a mate also attracts the fish that eats you. Most reef animals live inside that trade-off permanently, and much of reef coloration is a compromise between being visible to the right audience and invisible to the wrong one. A circular polarization signal cuts the trade-off. To an animal without the machinery, the patch is unremarkable. To a stomatopod, it carries information. You get the reach of a conspicuous signal without the exposure. Now recall what these animals actually need to communicate about, and the hypothesis gets sharper. They live in a permanent housing dispute, resolved through bluffing, displays, and calibrated violence in an environment where misjudging an opponent is expensive and where fights are settled by assessment. A dedicated channel that other species cannot eavesdrop on, carrying information about identity, sex, or status, would slot directly into that system. But this is a hypothesis, and it should be labeled as 1. The function of circular polarization signaling in these animals has not been pinned down. Alternative explanations exist and remain live, including the possibility that polarization sensitivity improves contrast in murky water, which would be a straight visual advantage having nothing to do with communication. Comparative work across multiple species found that circular polarization sensitivity and the reflective signals themselves are not universal in the group. Some species have them. Some appear to lack polarization signals entirely and may rely on color instead. That patchiness is exactly what you would expect if the system evolved for particular reasons in particular lineages, and it is a caution against treating it as a defining trait of all mantis shrimp. What is not in question is the detection. The eyes read it. That much is established, and it changes the character of the animal in a way that is hard to shake off. Add up what that system is actually doing, and the word eye stops being useful. Depth from a single lens. Recognition by sweep instead of by calculation. A spectral range that runs past the end of yours and gets sliced finer by pigment filters. And on top of all of it, 2 properties of light, orientation and rotation, that carry information your visual system throws away before it ever reaches you. That is not a pair of eyes in any sense you have experience with. It is a pair of independently steerable instrument packages, and every part of that instrumentation traces back to the same set of problems: range, because the strike cannot be corrected once it fires. Assessment, because the fights are settled by judgment. Recognition fast enough to matter in a habitat where hesitation costs you. Which brings the whole thing back to the weapon, and to a question that has been quietly waiting since the first frame. Not every mantis shrimp has a club. The animal described so far, the hammer, the cavitation, the shattered snail, is one branch of a larger group. Stomatopods are split, in the roughest terms, into 2 functional camps, and the second camp solved the same problem in a completely different direction. They are called spearers, and instead of a folded hammer, the terminal segment of the raptorial limb is elongated and lined with sharp spines. Rather than a club, it is a barbed harpoon. The behavior that goes with it is different. Smashers tend to live in rubble and hard substrate and hunt armored prey. Snails, crabs, hermit crabs, anything with a shell that needs breaking. Spearers tend to live in soft sediment. In burrows they dig themselves, and they hunt soft-bodied prey. Fish, shrimp. Things with no armor and one overwhelming advantage: they can leave. 2 totally different problems. Armored prey does not flee effectively, so the entire challenge is penetration. Soft prey has no armor at all, so the entire challenge is capture before escape. And here the numbers do something you would not predict. Measurements on a large spearing species, the zebra mantis shrimp, an animal that builds deep U-shaped burrows in sand and waits at the entrance, gave a mean peak strike speed of a couple of meters per second, with the whole strike lasting on the order of 25 milliseconds. Set that beside the smasher figures, smashing strikes in the range of 14 to 23 meters per second, complete in under 3 milliseconds. The spearer is roughly an order of magnitude slower, and its strike takes something like 10 times as long. At first glance, that makes the spearer look like the lesser version. It is not. It is the same underlying engine tuned for a different output. Both lineages use stored elastic energy released through a latch. What differs is what the mechanism is optimized to amplify. The smashing arrangement is built for force, converting stored energy into a violent, concentrated impact over a short distance. The spearing arrangement is built for displacement, converting stored energy into reach, throwing the limb out across a longer distance to intercept something that is trying to leave. That is the trade, and it is a real one in mechanics. You can amplify force or you can amplify displacement. The same energy can be delivered as a hard, short blow or a long, fast extension, but not both. A spearer that hit like a smasher would be useless. Shattering force applied to a fish accomplishes nothing if the limb does not reach the fish, and a fish at the entrance of a burrow has a fraction of a second before it is out of range. Reach is the whole problem. So the spearer built reach. And the evolutionary sequence behind that split is more interesting than the split itself because it does not run the way most people would guess. The intuitive assumption is that the hammer came first, that the club is the ancient primitive tool, and the spear is a refinement. Analyses of stomatopod evolution point the other way. The spearing form appears to be the older condition, and the smashing appendage arose out of it later, with estimates placing that origin roughly in the Cretaceous, on the order of 120-odd million years ago. Those dates come from molecular work combined with fossil calibration, and like all such estimates, they carry error bars rather than certainty. But the ordering is the point. The hammer is a modification of the harpoon, which means the smasher's whole world, cavitation, ceramic coatings, phononic filtering, Telson sparring, the housing shortage in coral rubble, is the downstream consequence of one lineage's limb-changing shape and becoming able to break things that were previously off the menu. And the group itself is far older than either version of the weapon. Push back through the fossil record, and stomatopods run deep. Early relatives appear in Carboniferous deposits with well-preserved specimens from the Mazon Creek fossil beds of Illinois, dating to somewhere around 307 million years ago, and related forms slightly older. Molecular estimates for when the crown group split from its nearest crustacean relatives run back into the range of 340 million years. Raw dates go numb fast, so translate them. 300 million years ago, the land was dominated by forests of plants with no flowers. The first reptiles were recent. Dinosaurs did not exist and would not appear for another 70 million years. The continents were assembling into a single landmass. Nothing you would recognize as a modern coral reef was in place. The animal lineage was already there, but, and this is where the fossil evidence has to be handled honestly, the early members were not the animals we have been describing. The Carboniferous forms had raptorial front limbs, yes, and they were clearly predators. But those limbs were more generalized, grasping appendages rather than the extreme folded, spring-loaded, latched machinery of a modern stomatopod. They look, in reconstruction, more like a shrimp with unusually equipped front legs than like the thing sitting in a coral cavity today. The spring came later. The hammer came later, still. And there is a hard limit to what any of this can tell us, which should be said out loud rather than glossed over. Fossils preserve shape. Shape constrains function, sometimes tightly. A joint that cannot bend a certain way tells you the animal did not bend it that way. But behavior is a different order of inference. A fossil limb can show whether the geometry for elastic energy storage was present. It cannot show whether the animal defended a burrow, whether it sparred with its own species, whether it displayed before fighting, or what its eyes could resolve. Those come from living animals, and they can only be projected backward with caution. So the honest picture is a shape with a long history and a behavior with a short one. We can say the body plan is ancient. We cannot say the ritual is. What the fossil sequence does give us is a chain of pressure, a generalized predator with grasping limbs, a gradual sharpening toward the spearing form, tuned to reach, then in one branch a rearrangement toward force, the limb folding, the terminal segment thickening into a heel, the spring stiffening, the latch tightening, and with that rearrangement, access to an entire category of prey nothing else in the neighborhood could open, which is exactly the kind of change that reorganizes a lineage. New prey means new habitat, because armored prey lives on hard bottom rather than in soft sediment. New habitat means new shelter requirements, because hard bottom offers cavities instead of dug burrows. New shelter requirements mean competition for a limited resource. Competition means fighting. Fighting with a shell-breaking weapon means either mass casualties or a ritual system to prevent them. You can watch the entire behavioral world of a modern smasher unfold from a change in the shape of one limb segment. It is a strange thing to watch happen. A change in the shape of one joint propagating outward through ecology, until it arrives at something that looks almost like etiquette. And the spearers, holding on to the older design, went in a direction that is arguably stranger, because the spearing lineages include some of the largest stomatopods and some of the most socially unusual. The zebra mantis shrimp digs a deep burrow in sand, a substantial excavation, U-shaped, with 2 openings, a piece of infrastructure that represents a large investment of labor, and it does not live in that burrow alone. Males and females of this species pair up, sharing a burrow, in an arrangement described as social monogamy. The female tends the eggs. The pair maintains the tunnel. Divers who return to the same patch of sand find the same pair at the same burrow entrance across long stretches of time. Now ask why. Because monogamy in a crustacean is not the default, and it demands an explanation. The explanation researchers point to is the burrow itself. When your shelter is a large, costly, laboriously constructed structure that cannot easily be replaced, the calculation changes. Abandoning it to search for mates is expensive. Defending it is worth a great deal. 2 animals maintaining one high-value structure, with one able to forage while the other guards, is a better arrangement than either could manage alone. The pair bond is not sentiment. It is co-ownership of an asset neither wants to lose. Figures get quoted for how long these pairs stay together and how long the animals live. Numbers running into decades. Those figures should be treated as soft. Long-term observation of individual invertebrates in the wild is genuinely difficult. Marking is hard. And much of what circulates comes from limited observation rather than systematic study. What can be said with more confidence is that these are long-lived animals by crustacean standards, and that stable pairing at a single burrow is a real, repeatedly observed pattern. Set the 2 side by side, and the contrast is hard to look away from. The same order of animals contains a solitary, permanently territorial smasher that spends its life fighting neighbors over holes in rubble, and a spearer that shares a home with the same partner for years. Same fundamental machinery, same spring, same latch, same eyes, different substrate, different prey, different shelter economics, and therefore a completely different social life. The environment wrote the behavior. It always does. That is the part that gets lost when an animal becomes an internet character, and it is the right place to come back to the first word in the promise, because there is a correction owed there too. Fastest strike in the ocean. That claim has been sitting unexamined for a while now, and it does not survive contact with the full comparison. Look outside the water, and the mantis shrimp loses. Several arthropods use the same fundamental trick: slow muscle, elastic storage, latch, explosive release, and some of them beat it outright. Trapjaw ants slam their mandibles shut at speeds that were, for a period, the benchmark for extreme animal motion. Then measurements on Dracula ants, which load their mandibles by pressing the tips together and letting one slide across the other in something mechanically similar to snapping your fingers, produce numbers considerably higher, peak mandible speeds around 90 meters per second, with the whole motion complete in as little as 23 microseconds. 90 meters per second is roughly 4 times the peak speeds recorded for smashing mantis shrimp strikes. 23 microseconds is a strike more than 100 times shorter in duration. So the mantis shrimp is not the fastest animal movement known. It is not particularly close. That should feel like a demolition of the whole premise. It is not, and the reason it is not is the entire point. The ant is moving through air. Air is roughly 800 times less dense than water and vastly less viscous. A mandible snapping shut in air is fighting almost nothing. The energy budget goes almost entirely into moving the mandible, and the mandible barely disturbs the medium it passes through. Put that same motion underwater, and the accounting collapses. Drag rising with the square of velocity. Added mass. The water that has to be accelerated along with the limb, which effectively makes the limb heavier the faster it moves. Energy bleeding out into the fluid at every instant of the motion. And then, past a certain speed, the fluid stops behaving like a fluid at all and starts tearing open into vapor cavities that slam shut against the very structure that created them. An ant's mandible does not have to survive its own shockwave. A mantis shrimp's club does, every time, for years. So the honest version of the superlative is narrower and better than the loose one. This is not the fastest movement in nature. It is something more specific and considerably harder. Among the most extreme accelerations achieved underwater, sustained repeatedly by a structure that has to withstand the destructive consequences of its own speed and remain functional afterward. Speed alone is not the achievement. Speed that does not destroy the animal producing it. That is the achievement. And there is one more constraint hiding in this comparison, which explains why you have never seen a large animal do anything remotely like it. Size punishes this strategy, and it punishes it fast. Scale an animal up, and the parts of it do not grow at the same rate in terms of what they can do. Mass climbs with the cube of length. Cross-sectional area, which is what determines how much force a muscle or a spring can deliver, climbs with the square. So a body twice as long is 8 times heavier, but only 4 times stronger. And every time you enlarge, the ratio of what you have to move to what you have available to move it gets worse. Elastic energy storage runs into the same wall. A spring stores energy according to its volume, so a bigger spring stores more. But a bigger limb also has more mass to accelerate, and it has to be accelerated through more water, and the drag on it scales with area while the energy available scales with volume in a way that does not rescue you once the speeds get high. Beyond that, big structures have longer natural timescales. A large spring simply cannot unload as quickly as a small one, because the material has to physically move further for the release to complete, and material can only move so fast. There is also the latch. The whole system depends on a small structure holding a large stored load and then getting out of the way in microseconds. Scale that latch up and it becomes a large structure holding an enormous load, which must now move a greater distance to release, taking longer, during which the energy is already leaking into the motion in a less controlled way. The precision that makes the mechanism work degrades with size. This is why the roster of extreme spring latch performers reads the way it does. Ants, frog hoppers, small shrimp, fungal spore launchers, plant seed-throwing mechanisms, cyanidarian stinging cells firing in microseconds. Everything on that list is small. Some of it is not even animal. The mantis shrimp sits near the upper end of what a body can be and still play this game. That, more than any single number, is what makes it remarkable. It is not a small mechanism. It is roughly hand-sized, in some species larger, operating at accelerations that physics generally reserves for things you need a microscope to appreciate. All of which drags the question back down to the ocean floor, and to what this animal actually is inside its ecosystem. Because the reputation says apex predator, and the reputation is, again, doing something misleading. A smashing stomatopod is a specialist, and specialists are narrow by definition. The club opens hard-shelled prey, that almost nothing else in the immediate neighborhood can open. That is a genuine ecological position, an animal converting armored, otherwise protected biomass into available food. Snails that had solved the predator problem by growing a thick shell find that the solution has stopped working. But a specialist is also constrained by its specialty. That weapon is expensive to build, expensive to maintain, useless during molt, and precisely tuned to a category of target. It does not make its owner invulnerable. It makes its owner very good at one job. And plenty of things eat mantis shrimp. Larger fish take them. Octopuses hunt them, which produces a genuinely tense encounter, since an octopus is soft, flexible, intelligent, and capable of approaching from angles the stomatopod cannot easily defend, while the stomatopod holds a weapon that could end the fight in one motion. Reef predators find them when they are exposed, when they are moving between cavities, when they are freshly molted, when they are foraging. This is what the burrow is for. Not comfort, not territory in the abstract. The burrow is a solution to being edible, so the correct picture of a smasher's life is not a rampaging destroyer. It is an animal that spends the overwhelming majority of its time inside a hole, with only its eyes exposed, watching, waiting for the right target to walk past the entrance, defending the hole from others of its kind, retreating deeper when a shadow crosses, emerging briefly and carefully to move between cavities or to forage, and returning as fast as it can. The weapon does not define the life. The hiding does. And there is a detail here that finally explains something a lot of people have heard secondhand and never had a reason for. Anyone who has kept marine aquariums has heard the stories. Mantis shrimp arrive by accident, hidden inside pieces of live rock, and are discovered later when fish start disappearing or when a sharp clicking sound starts coming from the tank at night. And there are long-standing reports among aquarists of these animals striking aquarium glass. Take that at the level it deserves, as a widely repeated observation among people who keep them, not as a controlled measurement, and think about what it actually implies. Aquarium glass is not weak, but glass is the textbook example of a brittle material, extremely strong under steady compressive load and vulnerable to exactly the kind of sharp, localized, high-rate impact that starts a crack and then drives it, which happens to be the precise failure mode this animal's entire weapon is optimized to produce. The animal is not choosing to break out. There is no plan. What is happening is that a territorial, cavity-dwelling predator has been placed in a transparent box, and it can see through the walls of its burrow to things it cannot reach, and it responds the way it responds to anything it wants to displace. The mismatch is not aggression. It is an animal running its normal behavior against a material that behaves like prey armor, and the clicking that gives them away in the dark is the other half of it. That sound is the strike, the impact, and the cavitation collapse, arriving as a sharp report. In the wild, that report is part of the reef's soundscape, one of countless small, sharp noises in an environment far noisier than most people imagine, which introduces a cost the animal cannot avoid. Every strike is an announcement. The same physics that generates the second hit also generates a loud acoustic signature. There is no quiet version of this weapon. To use it is to broadcast your position to anything listening. For an animal whose survival strategy is fundamentally about not being found, that is a real tension. And it fits with everything else about how the strike is used. Sparingly, from cover, aimed, with the animal returning immediately to concealment. None of which is how the story gets told. The version that circulates is a highlight reel. Punch, shattered glass, bullet speed, boiling water, a rainbow of impossible colors. Every one of those claims has a real phenomenon under it, and every one of them has been flattened into something that misses the actual interest. The punch is not a punch. It is a stored energy release through a latch, which is a fundamentally different kind of event. The bullet comparison is about acceleration and is species-specific and lab-measured, and it becomes far more impressive, not less. Once you add the medium, it happens in. The boiling water is real cavitation with measured forces, and the light is the part that should carry a caution rather than a headline. The rainbow is, by the best behavioral evidence available, close to backwards. Strip the exaggerations out, and what remains is stranger than what was removed. That is the part that keeps happening with this animal. Every correction makes it more difficult to explain, not less. And there is one more layer to it, sitting underneath everything discussed so far, that has been implied but never stated directly. All of this is being run by a nervous system that fits inside a crustacean. Consider what that system is managing. 2 eyes, each independently steerable in 3 rotational axes, each producing depth information on its own, each dragging a scanning strip of a dozen-plus spectral channels across the visual field, while simultaneously reading the orientation and rotation of light waves. A weapon that must be loaded over a comparatively long period, held under load, aimed with precision, and released at a moment determined by the position of a moving target. A behavioral repertoire that includes threat display, deception, opponent assessment, escalation decisions, and, in some lineages, long-term pair maintenance, spatial knowledge of a home cavity, and the surrounding terrain, including routes back to it. None of that requires a large brain in the vertebrate sense. It requires the right architecture. And the scanning model for Creek color processing is a good illustration of the general principle at work here. The animal is not solving problems with more computation. It is solving them with better arrangements, so that less computation is needed. That is the same design philosophy as the club. Not a stronger material, a smarter stack of ordinary ones. Not a faster muscle, an ordinary muscle plus a spring. Not a bigger visual brain. An eye that moves in a way that makes the brain's job smaller. Over and over, this animal answers a hard problem by changing the structure of the problem rather than by brute force, which is exactly what makes the remaining unknowns so uncomfortable, because we can describe the machine and still not fully explain how it holds together. Take the release itself. We know a latch exists. We know small hardened structures hold the loaded limb and that something disengages them. What is genuinely difficult is the timing. The energy stored in that spring is large relative to the size of the structures containing it, and the release has to happen in a window measured in microseconds, cleanly, without the latch surfaces grinding, without partial release, without the whole assembly shifting under load. In engineered devices, the release mechanism is usually the first thing to wear out and the hardest thing to make reliable. This one goes through thousands of cycles across a lifetime, in seawater, without being serviced. Then the energy accounting, which is messier than any summary makes it sound. Energy goes into the spring. Energy comes out as motion. But the fraction that goes into useful limb rotation versus what bleeds into the surrounding water, into vibration in the animal's own body, into the acoustic pulse, into heat in the deforming tissue. Those are hard quantities to pin down in a living animal, and the numbers depend on the size of the individual, the temperature of the water, the condition of the exoskeleton, and how recently it molted. Physical models built to imitate the mechanism have been used precisely because getting clean measurements from the animal is so difficult. Then the damage question, which is the one that keeps circling back. The club is built to take damage in a controlled way. Fine. But something has to determine when the accumulated damage is enough. A crustacean molts on a cycle, and that cycle is governed by growth and hormones. Does the state of the weapon feed into that at all? Does an animal with a badly chipped club behave differently? Strike less, choose softer prey? Avoid contests it would otherwise enter? Those are answerable questions, and they are not fully answered. Then the tissue behind the armor. Whether the wave filtering effect works inside a real, three-dimensional, water-filled limb as cleanly as it does in a simplified model is a question that will need experiments nobody has run yet. Then the vision, where the uncertainty runs deepest. We know what the receptors are. We know what wavelengths they respond to. We know the behavioral discrimination results, and we know the scanning proposal that came out of them. What we do not have is a full account of what the brain does with the signal. How a pattern spread out in time becomes a decision. Where in the nervous system that conversion happens, and whether the leading model is the whole story or one component of something more layered. And the polarization channel, where we can describe the hardware in detail and still cannot say with confidence what it is being used for. That combination is what makes this animal genuinely unsettling rather than merely impressive. Not that it is powerful, that it is legible only up to a point. We can measure the forces, we can slice the club and map every layer. We can record from individual photoreceptors, and we still cannot give a complete account of what it is like to be one, or of how the parts we have cataloged combine into the behavior we watch. The pieces are known. The assembly is not, which is a strange thing to say about an animal you can buy, keep in a tank, and film from 6 inches away. So rebuild the moment properly, because everything up to here is mechanism. And mechanism only means something once you put it back inside a life. Picture the reef at the end of the day, not the postcard section, the rubble field, past the living coral, where broken pieces of old skeleton pile up in a rough slope. Sand between the fragments. Low light, coming in flat and going orange as the water strips the red out of it with depth. Somewhere in that pile is a hole about the width of a finger. Inside the hole is an animal that has been there since morning. It has done almost nothing all day. Metabolically, this is the cheapest possible existence. A body at rest in a stable temperature, moving only enough to keep water passing over the gills. Occupying a cavity is not idleness, it is the strategy. What is exposed at the entrance is a pair of eyes on stalks, and the eyes are not still. They rotate, tilt, roll, sweeping the midband across the scene in a pattern of movements that would be pathological in a vertebrate and are routine here. Every fragment of rubble in view is being scanned, sampled across a dozen spectral channels, read for polarization, and ranged for distance by each eye independently. Something moves at the edge of that field, a snail working across the sand, carrying an armored shelter that has served its lineage for a very long time. Everything in the animal reorients. The eyes converge. Distance is measured, twice redundantly. Whether this target is worth a strike is being decided against a set of costs the animal never has to think about consciously. The energy of a loading cycle, the wear on a weapon that cannot be repaired, the noise of the impact and whatever might hear it, the exposure of leaving the cavity. Then the arm cocks. Muscle contracts over a comparatively long interval, compressing the saddle, ceramic layer taking the squeeze on the outside, fibrous layer taking the stretch underneath, the exoskeletal linkage folding into its loaded configuration. The latch takes the load. From outside, all you see is an animal that has gone tense and very slightly compressed, and then it holds, loaded, aiming. That pause is the strangest part of the whole sequence, and it is the one nobody puts in the highlight reel. There is a moment, brief but real, where this animal is standing perfectly still with a substantial amount of elastic energy stored inside a structure a few millimeters across, held in place by a latch, waiting for the target to arrive at the position the geometry has already been set for. Nothing about the outside of the animal indicates the state it is in. The snail keeps walking. The latch releases. And now the timescale changes so completely that everything human about the description breaks down. Under 3/1000ths of a second for the entire motion. The club rotating out and forward, water piling up in front of it and tearing open behind it. Impact. A peak force in the hundreds of newtons, delivered by an animal weighing a fraction of what a hand weighs. The shell responding the way brittle materials respond, with cracks initiating at the point of contact and beginning to run. Then, roughly 400 microseconds later, before the first event has finished, the second one. The vapor cavity slamming shut. A second force peak. Energy dumped into the same cracks that are already open. Inside the club, the same impact is traveling backward, into the mineral coating, which does not dent, into the herringbone layer, where the cracks that form are immediately turned sideways and stopped, into the graded spiral region, where the vibration is scattered and filtered before it can reach living tissue. The whole event is over before the animal's nervous system could possibly have influenced any part of it. And the sound of it travels out across the rubble field, and everything with ears has just been told where the animal is. The shell does not open. It is damaged, at the edge of the aperture where the blow landed, and that is all. So the animal turns the snail, sets it again, and reloads. Slow muscle, compressed saddle, latch, hold, aim. And it does it again. That is the part the highlight reel cuts. What follows is not an execution. It is a job. Repeated across minutes, blow after blow onto the same chosen weak point, each one costing a full loading cycle, each one carrying another impact and another cavitation collapse back into the club, until the shell finally gives up a hole large enough to reach through. Then the ordinary part, an animal eating. It is the same in every ecosystem. The spectacular part is measured in microseconds, and the rest of it takes a while. Then it goes back in the hole. That is the loop. That is the entire life. And every element of the machinery we have gone through exists to make that loop repeatable, to let the animal do it again tomorrow and next month and next year, without the weapon failing, without the arm shattering, without giving away the burrow, and without being eaten during the intervals of exposure, which is why the framing of this animal as a monster misses so badly. A monster does not have a maintenance schedule. And there is one more thing about that snail because it points at something larger than either animal. A shell is not a decoration. It is an enormous investment. Building one costs minerals, costs energy, costs growth that could have gone into reproduction, and costs mobility for the rest of the animal's life. Nothing pays that price without a reason, and the reason is always something trying to get inside. Hard-shelled marine invertebrates carry armor because being crushed and being drilled are ancient, persistent threats. Shell-breaking predation is one of the long-running pressures in marine ecosystems, and armor is the standard reply. Thicker walls, reinforced apertures, sculpture and ribbing that resist point loads, shapes that are hard to grip. That sets up the relationship properly. The Mantis shrimp club is not an arbitrary weapon that happens to break shells. It is an answer to armor. A mechanism aimed at the specific failure mode of the specific defense that most of its prey has invested everything in. And note what kind of answer it is. Not more force in the ordinary sense. A crab uses more force, a slow, powerful, sustained squeeze from a large claw, which works but requires the crab to be large and to keep hold of the prey while it works. The stomatopod solution is different in kind. It applies less total effort, but it applies it fast, and it applies it twice in immediate succession. And it exploits the fact that brittle materials fail by crack propagation rather than by being overwhelmed. Armor that would take a very long time to crush can be broken in microseconds by something that understands cracks. That is the thing about the whole design that is easy to miss under the spectacle. This is not raw power. Everything about the animal is leverage, using timing, geometry, material arrangement, and the properties of the target to accomplish what brute force could not. And every part of that has to be built from scratch by an animal that starts out as almost nothing. Because these animals do not hatch as miniature versions of the adult sitting in a hole. Stomatopods begin life in the plankton as larvae, drifting in the water column, transparent or nearly so, small enough that the world they experience is governed by rules the adult will never encounter again. At that size, water stops behaving the way it does for the adult. The smaller you are, the more viscosity matters relative to momentum, and the less you get to coast. Drag that an adult can ignore becomes the dominant fact of your existence. Everything about locomotion, feeding, and escape operates under a shifted balance of forces, and these larvae are predators already. They have functional eyes and raptorial appendages, and they hunt in the plankton, in open water with nowhere at all to hide. Which raises an obvious question about the spring. Does a millimeter-scale larva have one? Or does the whole elastic mechanism only switch on once the animal is large enough to need it? Research into how these mechanisms scale during development has gone after exactly that, examining strikes in larval stages rather than assuming the adult answer applies. The general point that comes out of that line of work matters more than any single number. The elastic system is not simply a scaled-down copy of the adult version. Spring-driven mechanisms behave differently at different sizes, and understanding when and how the machinery appears during a life cycle is a live question rather than a solved one. So even the beginning of this animal is unfinished business. Then they settle, and everything changes at once. A drifting animal has to find substrate. A larva that lands in the wrong place, open sand with no cavities, an area already saturated with adults, the wrong depth, does not get a second attempt. And from that point, the animal enters the cycle that will govern the rest of its life. Grow. Outgrow the shell. Molt. Harden. Grow again. Every one of those molts is the vulnerability window all over again. Every one of them is a period of days during which the weapon does not work, the armor does not protect, and the entire survival strategy reduces to hiding in a cavity and hoping nothing comes in. Every one of them requires a cavity large enough to accommodate the new, slightly larger body, which means the housing problem is not solved once, but reopened at every stage of growth. An animal that has held the same hole for months may find after a molt that the hole no longer fits. Then it has to go out and take one from someone else, which means contests, which means displays and Telson sparring, which means risk. Look at what that produces over a lifetime, and the emphasis shifts entirely. The strike, the thing the animal is famous for, occupies a vanishing fraction of its existence. Add up every strike a smasher makes in its life, and you are summing events of 3 milliseconds each. Even at thousands of strikes, the total time spent striking is measured in seconds. Everything else, years of it, is sitting in a hole, watching, and periodically going soft and defenseless while the machinery is rebuilt. That is the survival story underneath the monster story. The weapon is not the hard part. The intervals are the hard part. And the weapon has to be regrown, at full quality, again and again. Every molt means laying down a new mineral coating, a new herringbone layer, a new graded spiral, all of it correctly ordered, in an animal that is at that moment soft and hiding and cannot feed effectively. The most sophisticated composite material anyone has found in a crustacean gets manufactured, from the inside, repeatedly, under exactly the worst conditions the animal ever experiences. Set that next to the biomimetics for a second. Prototype composites inspired by this club get made in controlled facilities, with defined fiber layups, cured under specified conditions. The animal makes its version in seawater, while starving, while hiding, and then immediately puts it into service against a snail. So build the picture back up now with everything in place, because the animal you started with and the animal you are looking at now are not the same creature. It is not fast. It contains something fast, released by a latch that took several 100 million years of lineage history to arrive at. It is not violent in the sense the word implies. It is territorial, and it lives inside a housing shortage that turned its weapon into a language. Displays, bluffs, measured blows on armor built specifically to receive them. It does not see a rainbow beyond human imagination. By the best behavioral evidence, by the best experience, it discriminates color worse than you do, and appears to have traded that precision for speed of recognition and a cheaper nervous system. Its eyes are not eyes in the way yours are. They are three-part optical instruments on independently steerable mounts, each capable of ranging on its own, reading properties of light, ultraviolet, linear polarization, circular polarization, that your visual system does not register. Its armor does not resist damage. It absorbs and redirects it, guiding cracks sideways and filtering the vibration of its own strike away from tissue that would tear. And it is periodically completely helpless. And it lies about that helplessness by performing a threat it cannot carry out. Every single one of those corrections makes the animal harder to explain than the myth did. And that pattern is the part that outlives the animal, because it is not really about mantis shrimp. The popular version of any animal is a compression. It keeps whatever is easy to picture and drops everything that requires holding two ideas at once. A punch is easy to picture. A latch releasing stored elastic energy through a folding exoskeletal linkage is not. 12 color channels sounds like more colors, because that is the obvious reading, and the obvious reading is wrong. The compressions are not lies exactly. Each one started from something real. They just lose the mechanism, and the mechanism is where the strangeness actually lives, which leaves the oldest part of the whole story still unaccounted for. The lineage has been running this experiment for an amount of time that does not fit comfortably in a human head. Try to make it concrete rather than numerical. The early relatives preserved in Carboniferous rock were alive in a world where the atmosphere had a different composition, where the continents were fusing into one mass, where insects grew to sizes that modern oxygen levels do not permit, and where the forests laying down the coal we would eventually dig up were still standing. There were no birds, there were no mammals, there were no flowers, there were no dinosaurs. There were already predatory stomatopod relatives on the seafloor, with raptorial front limbs, doing a version of what the animal in the rubble field does now. Between then and now, the ocean has been emptied and refilled more than once. The end Permian event removed the overwhelming majority of marine species. The end Cretaceous event took out the large marine reptiles and reorganized every food web in the sea. Between and around those, sea levels rose and fell hundreds of meters. Ocean chemistry shifted. Reef-building organisms rose, collapsed, and were replaced by different reef-building organisms. The group persisted through it, not unchanged. That is the important qualification. And it is where the fossil record forces honesty. The animals that came through were not the animals that went in. The spring evolved somewhere along that path. The smashing form arose, by current estimates, in the Cretaceous, which means the hammer is a comparatively recent addition to a very old body plan. The eyes we find so difficult to explain are the product of elaboration within the group over long stretches of time, and reconstructing when each layer of that complexity appeared is exactly the kind of question fossils are bad at answering. What survived was not a design. It was a lineage that kept generating designs. And that reframes the deep time part in a way that is more useful than simple awe at big numbers. The mantis shrimp is not ancient in the sense of being unchanged. It is ancient in the sense of never having stopped solving problems. The current version, hammer, ceramic coating, phononic filtering, scanning eyes, Telson armor, ritual combat, is the most recent state of an ongoing process, not a relic. So the animal in that hole is not a survivor from a lost world. It is the current edge of something that has been continuously in progress since before the first tree, which leaves the hardest thing to attempt, occupying the animal's position. Instead of describing it from outside, the light is going. Above, the surface is a shifting ceiling. Red has already been stripped out of the light by the water column, so most of what an ordinary camera would record is blue-green and flat. The stomatopod's version of the scene is not that. Start with the parts we can be reasonably confident about. Ultraviolet is present, filtered, sliced into separated bands by pigments sitting in the optical elements above particular receptors. So there is a whole region of the spectrum contributing information that produces no experience for you at all. Some marine organisms reflect and absorb ultraviolet in ways that make them stand out sharply in that range, while looking unremarkable in ours. Layer polarization on top. The water itself carries polarization patterns that vary with viewing angle and depth. Surfaces reflect it. Some animal tissues produce it. To an eye that reads orientation, a scene that looks uniformly murky can carry structure, edges, shapes, and contrast where none exists in brightness alone. That last point is one of the live hypotheses for why polarization sensitivity is useful here at all. In turbid water, it may pull contrast out of a scene that has almost none. Then add the rarest layer. Somewhere in that field of view, on the body of another stomatopod, there may be patches reflecting light that rotates as it travels, a signal broadcasting into a channel that most of the reef cannot receive. Whether it is carrying information about sex, status, or identity remains unsettled, and not all species have it. But where it exists in plain view, unread by almost everything watching. And then, cutting across all of it, the movement. The eyes are not holding a steady image. The midband is being dragged across the scene in scanning sweeps, sampling as it goes, building recognition from sequence rather than from a computed picture. Whatever this animal experiences, it is probably not a photograph. It may be closer to a stream of matches. None of that is a metaphor for human vision. There is no version of it you can construct out of your own experience, because the input categories are different. This is not a richer version of your world. It is a different set of questions being asked about the same photons. And that is the honest limit of the whole exercise. We can list the receptors. We can describe the eye movements. We can characterize the signals. We cannot get inside it. And the parts we would most like to know, what the animal is actually resolving, what it is attending to, what any of it amounts to from the inside, are exactly the parts that no measurement has reached, which is where the strangeness finally settles. And it is not where you would have expected it at the start. The frightening thing about this animal was supposed to be the weapon. It is not. The weapon is the part we understand best. We can measure the forces, map the layers, model the spring, film the cavitation, and copy the architecture into composite panels. The frightening part is the eyes, because the eyes are watching in registers we cannot enter, running a form of visual processing we have not fully described, on an animal that has been refining that system since before anything on land had a backbone worth mentioning. And the only reason we know even that much is that people found ways to watch back. The difficulty there is not incidental. Almost everything described here happens either too fast to see, too small to resolve, or inside a structure that has to be destroyed to be examined. Consider the basic problem of measuring a strike. The event lasts under 3 milliseconds. To capture it usefully, you need thousands of frames per second, which means very short exposure times, which means you need an enormous amount of light on a subject sitting in water. And the subject is a shy animal that spends its life avoiding exposure and will not perform on request. Then you need it to strike something in a known position, at a known moment, within the narrow depth of field of a high-speed lens. Then measuring force, you cannot put a sensor on the animal. So the sensor goes in the target, which means building something the animal is willing to hit, instrumented finely enough to resolve two separate force peaks a few hundred microseconds apart, and stiff enough not to distort the measurement by flexing. The discovery that there were two peaks at all, and therefore that cavitation was contributing a second, independent blow, came out of exactly that kind of instrumentation. Without the temporal resolution to separate them, the whole event would have looked like one hit. Think about what that means. The second impact was not hidden in some inaccessible part of the ocean. It was hidden inside the resolution of the measurement. It was always happening. We simply could not see the gap between the two events until the equipment got fast enough. Then, the material. Mapping the club means sectioning it, polishing it, imaging it at microscopic scale, running mechanical tests on regions smaller than a grain of salt, and doing it across a structure whose properties change continuously with depth. And every piece of that work destroys the sample. And then, for the parts that cannot be measured in the animal at all, people build the animal. Physical models of the mantis shrimp mechanism have been constructed. Engineered spring latch devices that reproduce the geometry of the linkage and can be fired repeatedly under controlled conditions. The reason for doing that is not to make a novelty machine. It is that a physical model lets you change one variable at a time. You can alter the stiffness of the spring, or the geometry of the latch, or the mass of the limb, and see what happens to the output. You cannot do any of that with a living animal, because the animal comes as a package and does not offer adjustable parts. What those models are aimed at is a question the animal itself cannot answer. Which features of the design actually matter, and which are incidental? Is the extraordinary performance mostly about the spring, or mostly about the latch, or mostly about the geometry that connects them? For a system where everything is coupled, the only way to find out is to build a version you are allowed to break. That is a strange and slightly recursive situation. In order to understand a biological machine, people built a mechanical copy, and the copy is now teaching them how to read the original. And the same logic runs in the other direction, back into engineering. Spring latch mechanisms have become their own field of study, because the underlying principle, load slowly, store, hold, release faster than any motor could, is exactly what you need for small machines. A tiny robot cannot carry a powerful motor, but it can carry a spring and a latch, and then it can jump, or strike, or snap, with a power output that its power source could never have delivered directly. The animal did not invent that principle. Physics did. But the animal found an unusually complete version of it. And it found the version that works underwater, which is the hard case. There is a detail in all of this that should not slide past unexamined, because it says something about how often nature converges on the same answers. The mantis shrimp is not the only crustacean that weaponized cavitation. Snapping shrimp? An entirely separate group, small, common, living in burrows and crevices across warm, shallow seas. Solved a similar problem with a different piece of anatomy. Instead of a hammer, a specialized claw with an enlarged movable finger that is cocked open, held, and released. The claw slams shut and drives a jet of water fast enough to cavitate. The collapsing bubble is the weapon. Prey is stunned or killed by the shockwave rather than by contact. Two lineages, two different limbs, two different mechanical arrangements. Same underlying physics, discovered independently, And snapping shrimp are so numerous in some habitats that their combined snapping produces a continuous crackling that dominates the ambient sound of shallow tropical water, a sound loud enough to matter for sonar operators and to be a standard feature of any hydrophone recording made in the right places. That puts the mantis shrimp's acoustic problem in a slightly different light. In a reef environment already saturated with sharp clicks from thousands of small crustaceans, one more report may be less of a beacon than it would be in silence. That is speculation about a specific animal's exposure rather than an established finding. But the soundscape is real, and it is much noisier than the popular image of a quiet reef. What matters more is the convergence itself. When two unrelated lineages independently arrive at cavitation as a weapon, it stops being a curiosity of one strange animal and becomes evidence about the physics of living underwater. There is an accessible region of the design space where, if you can move a limb fast enough, the water will do part of the work for you. Evolution found it twice. So the mantis shrimp is not a freak. It is one solution among several to a problem the ocean poses to anything small, hungry, and living next to armored food. And once you see it that way, the pieces stop being a collection of separate marvels. They are one problem, answered in a dozen places at once. The problem is this: how do you release a large amount of energy very fast, underwater, repeatedly, for years, without destroying yourself in the process? Every part of the animal is a clause in that sentence. The slow muscle is there because fast muscle cannot deliver the power. The saddle is there because the energy has to go somewhere that will not buckle. Its ceramic over fiber construction is there because storing that much strain in one material would crack it. The latch is there because stored energy has to be held and then let go on command. The mineral coating is there because the striking face cannot be allowed to dent. The herringbone layer is there because a hard face on its own would shatter. The graded spiral underneath is there because the animal's own strike would otherwise reach tissue that cannot survive it. The eyes are there because a release that cannot be recalled has to be aimed before it fires. Pull any one of those out and the rest stops working. And the one feature everybody knows about, the strike, turns out to be the part that forced all the others into existence. The speed created the material problem. The material problem created the wear problem. The wear problem created the molt vulnerability. The vulnerability created the need for shelter. The shelter created the scarcity. The scarcity created the fighting. The fighting, combined with the weapon, created the need for ritual, for armor, for display, for assessment, and therefore for eyes capable of reading an opponent fast and accurately in bad water. Follow that chain in either direction, and you end up somewhere unexpected. Start at the punch, and you arrive at etiquette. Start at the eyes, and you arrive back at the punch. And it means the two words in the promise both survive, but not in the shape they arrived in: fastest. Yes? With the qualification that the record for raw speed belongs elsewhere, in air, to animals that never have to survive what they generate. What this animal holds is something harder to beat: extreme acceleration in the one medium that fights back hardest, delivered by a structure that then has to withstand the shock wave it created and do it again next week. Most violent. Yes, with the qualification that the violence is almost entirely regulated. The forces are real. The cavitation is real. The broken shells are real. But most of the aggression in this animal's life is directed at its own species, over holes in rubble, and it is delivered onto armor built specifically to receive it, in exchanges that function as measurement rather than murder. The most violent predator in the ocean spends a great deal of its violence deciding who gets to keep a hole. That is stranger than the myth. It is also considerably harder to fit on a thumbnail. And notice that every place this story corrected itself, it corrected in the same direction. We assumed the fast thing must have a fast muscle. We assumed the hard thing must be hard all the way through. We assumed more color channels must mean more colors. We assumed a lethal weapon must mean lethal fighting. Every time, the intuitive answer was the simple one, and every time the real answer was an arrangement, a structure, a sequence, a piece of geometry that got the result without the obvious ingredient. That pattern is not unique to mantis shrimp. It is what biology does. It routinely reaches outcomes by rearrangement rather than by force, and our instincts, built for a world of pushing and pulling at human scale, keep guessing wrong about it. So go back to the rubble field one more time. It is nearly dark now. The reef has changed shift. The daytime fish have gone into cover, and the night animals are coming out. The sand between the coral fragments is gray. Whatever red remains in the world above has been gone from this depth for a long time. There is a hole in the rubble about the width of a finger. Nothing about it looks like anything. There are thousands of holes like it in a few square meters of reef, and almost all of them are empty. In this one, something is home. You would not see the body. The body is back in the dark, folded, still doing the cheapest thing it can do. What is at the entrance is a pair of eyes on stalks, and they are moving. Rotating independently, rolling, sweeping the narrow band of receptors across the scene in the constant scanning motion that never quite stops. Reading ultraviolet you cannot access. Reading the orientation of light waves you cannot detect. Measuring distance twice over, redundantly, on anything that moves. And folded underneath, held ready, is a spring made partly of ceramic, wrapped in a coating harder than most things biology bothers to build, sitting behind a latch a fraction of a millimeter across. Not loaded, not yet, but capable of being loaded in a fraction of a second, and of releasing everything it holds in less than 3/1000ths of one into a volume of water that will briefly stop behaving like water at all. The lineage that arrived at this was already old when the first forests were laying down coal. It has outlasted the emptying of the oceans more than once. It did not survive by staying the same. It survived by continuing to solve things. And the animal in that hole is not a relic of any of it. It is the current answer. Provisional, like all of them. If you were down there with a light, the beam would find those eyes, and they would find you back. Not with recognition, not with anything you could call attention in your own sense, but with a set of measurements being taken. Distance, size, movement. Whether this is a threat, a competitor, or something worth the cost of a strike, you would be scanned in registers you have no access to, by an instrument nobody has finished explaining, attached to a weapon we have taken apart layer by layer, and still cannot entirely account for. And then the light would move on, and the eyes would go back to watching the sand, and the hole in the rubble would look like nothing again. That is the part that should sit uncomfortably. Not that the animal is dangerous, it is barely dangerous to you at all. The unease is that something this thoroughly engineered has been sitting in shallow water the entire time. In reefs, people snorkel over on holiday, doing all of it in the open, in plain sight, while we walked past it for most of our history and got the story almost entirely wrong, starting with what we called it. It is not a shrimp. It is not a mantis. The name is wrong twice before it even reaches the animal. What is actually in that hole is a stored energy machine with a wear schedule, a housing crisis, a private language, and eyes that were reading the reef in channels we did not know existed until we thought to look. And it is still down there, in the dark, aiming at nothing yet,